Hello there. Welcome to GTB523, which is Techniques in Biomedicine Laboratories. This presentation will discuss about exploring new technologies in biomedical research. It also will discuss regarding innovation using human-based in silico and in vitro models for drug and device discovery. The main focus of this presentation is organs on chips, in which creating a living system to emulate human biology and disease. This presentation is based on an article journal by Benham et al. 2019. In this particular slide, it explains on what are organs on chips, which is basically generated using microscale engineering techniques. The principle behind organs on chips that is enable us to study the complex human physiology and pathology. The features of organ on a chip, which is similar to the size of an AA battery, and the function of organs on chips which related with perfused microchannels. Lungs on chips. The development of new solutions to treat pulmonary diseases has become today a goal of primary importance. Ha et al. Have managed to reproduce the function of an alveolo-capillary membrane, which is the smallest functional unit in the lung. That organ is vital to test new medicine since it constitutes the physical barrier between the body and the external world. Here is the main advantage of lungs on chips compared to traditional cell culture, as the following example shows. Liver on chips. Liver cells are among the most difficult to keep alive in a petri dish. Young et al. Proved that when mesenchymal cells from human placenta were put together with liver cells, the liver cells multiplied and their metabolic activity surged. Chen Ta et al. Have recreated on a microfluidic chip, which is the functional unit of the liver, by dielectrophoresis. This lobule consists in liver cells and endothelial cells. The main advantage of the liver on chip is its capacity to replicate microscopic details for more reliable tests. Brain on chips. The advantage of brain on chip over other in vivo and in vitro models comes from more realistic dimensions and geometry. Douth et al. Have also managed to build a multi-region brain on chip that could help model neurodegenerative diseases very close to the in vivo models. There are four main applications in organs on chips. They are use of low technology readiness, access to personalized medicine, the battle against cancer, and anti-aging research. The challenges and limitation of organs on chips are relative size of all organs on chips, the quantity of fluid inside the chips, and finding a universal substitute for blood. However, there are ways and strategies to improve this matter, which is work alongside investigators and industry in the development and validation processes and aim for collaborations. In conclusion, systematic use of organs on chips would help the pharmaceutical industry save time and money, and would as well limit the breeding of animals destined to clinical testing. The chips could also become formidable research accelerators, since they could allow to conduct many trials much more rapidly early in the research phase. This presentation is based on these following references. With that, thank you for listening for this video presentation.